Okay, so hopefully from the video you can see just how important enzymes are and how every reaction that's going on in our body requires an enzyme of some sort. Now, the important thing to remember with enzymes is that they aren't necessarily required. Like a reaction in our body will happen without an enzyme, but the enzyme actually speeds the reaction up. So to define an enzyme, it is a protein. So this is, I'm going to highlight the important things to keep in mind about enzymes. So first of all, enzymes, they're all proteins, so they're made up of amino acids. They catalyze or they speed up chemical reactions, but they're not used up, so they can be reused over and over again. And how they speed up a reaction is, if you think about a chemical reaction happening, you need a certain amount of energy to make that reaction happen. And what an enzyme does is it lowers the amount of energy needed, so it allows it to happen easier. And the other thing about enzymes, not always, but most of the time, their name will end in A's, and we'll look at a couple of examples. Okay, so what does the process look like? There are things called substrates. This is a reactant that the enzyme binds to or acts on. And it binds to a particular site called the active site. So these are important terms here. And it's usually like a pocket, a groove, or a certain sort of shape that the substrates can fit in, or the enzymes can fit into. When they're attached, they call it an enzyme substrate complex. So there's a little visual down here to kind of show you what happens, and then I'll put a real life example to it. So if you have an enzyme, here's what we call their active site, where there's sort of a groove here, and then you have some sort of substrate, so something that needs to go through a reaction. That substrate will have the same shape as the enzyme's active site, and it will be specific to that substrate. It will come in, bind, completing the enzyme substrate complex, and then either one of two things can happen. If this is a complex sort of molecule that needs to be broken up, or a catabolic reaction, then the enzyme will act on that and make that happen. So see, this was a two units joined together through a reaction, and the purpose of this reaction was to break them apart, so a catabolic reaction. Keep in mind that the reverse can also happen. You can have two single units that need to be joined together, and you might have an enzyme involved in that process as well. So just to put sort of a real life example into play here, if we were to use these as an example, so lactose is a molecule found in milk. So we could say, oops, I lost my lactose there, hold on. Okay, so if we were to think about lactose, so lactose is a molecule found in milk. And lactose is actually made up of, so remember ose tells you that it's a sugar. And because there's two units bound together, we can call lactose a disaccharide. So this is an example of a disaccharide. It's a molecule found in milk. And if you've ever heard of people who are lactose intolerant, it's this reaction that I'm about to show you that they're not able to do. And it's usually because of their lacking this enzyme. So here's an enzyme called beta-galactosidase. Remember, most enzymes end in A's. Most sugars end in ose. So this enzyme here, beta-galactosidase, is what helps break lactose down when people drink milk or have dairy products. So somebody who's lactose intolerant don't have this enzyme, or they have some sort of poor functioning with that enzyme. And so if you think about this process going on, the purpose of beta-galactosidase is to break down lactose into glucose and galactose. So lactose is made up of the two simple sugars called glucose and galactose. And in order to break it down when you drink milk or have any dairy products, it requires this enzyme here. Okay, so how do we control all the enzyme reactions going on in our body? There's three ways that enzymes can be affected, either stopped, sped up, or changed in some way. So there's the first two I'm going to reveal right together because they're sort of the opposite of each other. So one way an enzyme's action can be affected is competitively, meaning that there's some other molecule in our body that's competing for the same site that it's trying to bind to. And if there's some other molecule that has the exact same shape, then it can bind to that site, and basically what happens is it gets blocked. So competitive inhibitors block the site and prevent the real enzyme from binding to it. Now, the word inhibit is just another fancy term for it's stopping something from happening. So if we're inhibiting this from happening, we're stopping it. We can do it competitively by blocking it, meaning that there's some other molecule that's got the exact same shape and it's mimicking it, or non-competitive. There's some substance in our body that maybe just changes the shape of the enzyme, and now it can't bind to that site, because remember, those sites are very specific in terms of its shape. So if there's another molecule in our body that changes the enzyme shape, then that enzyme can no longer bind to that substrate. And then the third way is through something called feedback inhibition. Feedback means that it's communicating to the beginning of some sort of reaction or cycle. So for instance, if we have a multi-step reaction that's going on in our body and it's producing certain molecules, maybe what happens here is the production of one specific molecule. If the concentration of that molecule becomes high, or it makes a lot of it, it's a signal to the beginning of that reaction that it needs to slow down. So something being produced later on in a reaction gives the signal that that reaction needs to slow down because we have too much of what we're looking for, or we have enough. That's called feedback inhibition. Now, some real life examples. I'm sure you've heard of cyanide before. It's a strong poison. Cyanide competitively inhibits, so it competes and it binds to a site during the process of cellular respiration. So cellular respiration is basically every living organism goes through a process of cellular respiration. For us, we take in oxygen. Through cellular respiration, we can breathe out carbon dioxide. And the second one is antibiotics. Most antibiotics basically are inhibiting the process of some sort of reaction that requires an enzyme. So for instance, penicillin, it stops the bacteria that are in your body from being able to make it a component of its own cell membrane. So that's something that helps a bacteria sustain its own life. And antibiotics, something like penicillin, will stop the bacteria from doing that, and eventually the bacteria will die off. Now, one thing to keep in mind is that enzymes are very sensitive, and they need a specific environment to work in. And so if we take our body, for instance, we have a certain body temperature, and the pH in our body is different depending on where you are in your body. Now, how does this affect enzymes? Let's start with temperature first. If we think of temperature, most chemical reactions will actually increase or speed up with an increase in temperature. So if we increase the temperature, generally that actually helps a reaction along. When it comes down to an enzyme, it does do that, but there's a certain temperature it reaches, and it all depends on the specific enzyme we're talking about. But every enzyme will increase its activity with a certain increase in temperature, but then every enzyme will reach a certain spot where it actually decreases it. And what's happening is, I'll explain in a minute. So you have an optimum area of temperature for each enzyme, okay? And you also have an optimum pH for each enzyme. You guys might be familiar with these enzymes, these two here, pepsin and amylase. Pepsin is an enzyme that's found in your stomach. And if you recall, your stomach is made up of acid. So your stomach makes an acid with a very, very low pH. Some enzymes would thrive in that environment. Other enzymes are not going to do so well. Pepsin is designed to be very active in a very low pH. So that's why it's found in your stomach. Amylase, on the other hand, that's an enzyme that's found in your mouth. And we know that the pH of our mouth is not really, really low. It's close to a neutral. So depending on the enzyme, they also have an optimum pH that they work in. Well, what happens if they're outside of their optimum pH? This is a really important term. Something called denaturation. Denaturation is basically the loss of structure and function of a protein. Remember when we were talking about proteins, I said that their entire shape determines what they do. And if their shape changes, then they can no longer do what they're supposed to do. And if we have a change in either pH or temperature, then what's happening is that enzyme changes its shape. And because an enzyme is a protein, it can no longer do what it has to do.